So good morning. I must draw the short straw to speak first in the program 2 days running. I'm going to have a word with the program organizers about this. Yesterday I did a review. Today I just want to give you really a progress report on some of the work we're doing on online water quality measurement for the drinking water system. Just to remind you, we didn't start out so much with this in mind. When we first came here in Sensam Phase 1, we were primarily focused on wireless sensor networks, primarily interested in hydraulic parameters for leaks and burst detection. But part of the game plan when we arrived was to move towards water quality, the logic being that the setup we had from the first phase of Sensam would form the basis for monitoring water quality online. And it turns out the topic of measuring water quality online is extremely difficult because of course, water quality can be measured by sort of indicator parameters, but they don't give you very much information on whether water quality anomalies are potentially threatening health or other important features. So alerting people on water quality issues is much harder than detecting a burst where you know the pipe is broken and something has to be done. As we evolved this work into phase 2 of Sensam, we very quickly realized that most of the discussion should be around the microbial ecology or biofilms forming in water pipes. And I'll try and give you some reasons for this. When we first started working on this topic, we had sensors installed downtown Singapore. There was a test bed, which we had called WaterWise, and we thought that this was going to be the basis for doing all this validation and this work. As you'll see, we've had some challenges in doing this. You'll also see that one of the things we've focused on is a parameter to try and measure the growth rate of the biofilm. And for this, we teamed up with a startup company in Italy which produced a product called an Alvin probe, which measures bioelectrochemical signal. The final goal, though, of this work is to come up with reliable online sensors for water quality event detection, meaning events of significance. Why is all this important? Well, of course, drinking water is very heavily regulated for safety, for public health issues. In the US, at least, we have the Safe Drinking Water Act, which starts from the 1970s and has been revised more or less every 10 years. I don't know if there's a more recent revision. People study outbreaks of disease associated with drinking water systems. And you can see in the US over this period of time, there were recorded something in the order of 800 disease outbreaks. 0.6 million illnesses and something of the order of 80 deaths. That's the numbers that were quoted. These are hugely underreported. When you get gastrointestinal infections, you don't always report them to the hospital. But the thing I found very interesting is that most of the outbreaks that we're seeing in the drinking water systems are actually occurring because of things that are attributed to what's happening in the pipe network. And you can see this from this figure from NRC about 10 years ago. The blue is simply the number of outbreaks, health outbreaks in the system, which you can see are deteriorating or diminishing over time. But then you can see that the percentage due to distribution systems, in other words, the pipe network, are up around 60, 70%. In other words, most of the problems that people are reporting of public health issues relating to water quality are in the pipe network, not in the prior treatment, if you like. So this sort of final chapter of taking treated water from a treatment plant and getting it to the customer, it's that bit in between that somehow is letting us down. Now, this has all become very, very obvious in the US this last year, thanks to the city of Flint, Michigan, whose water looked a bit like this. I think probably you and I wouldn't drink the stuff. Very quickly, the testing of the water coming out of the taps has revealed the obvious, which is there's a lot of contamination in the water, in particular very high lead counts. In fact, you'll see if you can read this figure, this is the distribution. The green are obviously below EPA threshold. EPA threshold on lead is something like 15 parts per billion. And it goes up to an order of magnitude higher than that. And people and young children and so on who are particularly vulnerable to lead poisoning are very seriously affected by this and have been drinking this water for several years. The hidden part of the story, which is probably more pronounced in practical, immediate terms, is that there's been a very large increase in the occurrence of Legionnaire's disease. And Legionnaire's disease is linked to Legionella, in particular to Legionella pneumophila. And you can see that The city of Flint switched its water supply somewhere in 2014 and switched back again somewhere in 2015, and these are the occurrences of Legionnaire's disease. You can see they're correlated with this switch in water supply, and all of the suggestion is that this is linked to problems of Legionella buildup inside the water pipe somewhere. There are many unknowns in this system as to why we're getting lots of lead and why we're getting Legionnaire's disease. There's a lot of unknowns here. But I just thought I'd bring your attention, first of all, the source of lead. Where is the lead coming from? Well, it turns out Flint isn't supplying water in lead pipes. The lead pipe exists only at the household connection, and only in a few places. So it's unlikely just to be the corrosion of lead from lead pipes. More likely, it's associated with collection of rust in the water mains and lead being deposited inside the rust. And this is coming then from the water supply. There's more lead in the water supply. And the result, when they switched the water supply of having more corrosion, was that rust was picked up. And you can see the rust rather clearly in the color of the water. So they've moved, the release of lead is likely due to a change in more corrosive water source. The Legionnaire's disease, well, it turns out that these conditions are favorable or favor the creation or accumulation of Legionella in the pipe biofilm. But why I say it's significant, we've already had 12 fatalities from this spike. So relative to the incidents that have gone before, you can see this is a very significant event, and all essentially by switching a water source. So this has really got our attention focused on the whole issue of microbes in the water distribution system. Now, of course, in water treatment, you try very much to try and suppress pathogens and other things. Here in Singapore, Legionellosis is also prevalent. And the occurrence of Legionellosis in Singapore has been dropped by about a factor of 50 to 60% about 10 years ago by introducing chloramine treatment of the water. So this is what the public health agencies in Singapore tell us. So PUB, using chloramine treatment, is trying to suppress Legionella. The water pipe is not a sterile environment. So within the bulk water, of course, we have residual disinfectant to suppress the buildup of microbes, especially harmful opportunistic pathogens. We have a lot of other stuff in the microbial ecology in the drinking water. We have agents which bring about nitrification. Nitrification, or creation of nitrates, is also very harmful to humans, and in fact is a clear indicator of deterioration of water quality. We also have microbes that act to increase corrosion of pipes, of metal pipes. When we did our studies, you'll see we found Gallionella, which is very active in promoting corrosion of iron. So there are many adverse effects of the biofilms that form inside water pipes. We may have microbial pathogens, we may promote corrosion, and in fact, the thing that got us interested in this to start with is the probably most innocuous one, which is you put a sensor inside the water pipe and it deteriorates over time due to the biofilm formation. And the biofilm forms on the surface, but it also mixes with the bulk reaction. And one of the first questions is, are problems associated with bulk water correlated with the populations or the microbes that live in the biofilm? In many parts of the world, the bottom line is people don't drink the drinking water. In many cities in China, water is treated, but no one drinks it. And the implication here is that the microorganisms inside the pipes are what are preventing this. So trying to eliminate them is also a problem. Once they get created, it's hard to remove them, except by very extensive flushing or replacing of pipes. So these are the probes that we had in Singapore, in downtown Singapore, in our early development phase. This is an example, and it's just trying to show you we had a lot of biofilm on different substrate surfaces, on brass, on plastic, on stainless steel, and so on. So our initial approach to this problem was to turn around and say, well, we've got these probes, they're collecting biofilm, let's look at the properties of the biofilm and see how they correlate with what's in the bulk water. And later we decided that measuring biofilm growth rate would be a useful thing. And I should say this work was initiated with Masaki Kitajima, who's here today and is speaking next. And we found interesting things. We initially targeted assays for particular opportunistic pathogens. For example, This case is Mycobacterium. Mycobacterium is 150-odd species of bacteria. Most of them are harmless, although tuberculosis is a mycobacterium. This is a particularly interesting case because we expect it to be very resistant to the disinfectant, to the monochloramine that's in the pipes. And we find, in other words, Mycobacterium in many places within the pipe network. And you get some sense as to the types of testing we did from these studies. Each of the little diagrams here is showing us part of Singapore and showing us a distribution of measurements within the water pipe network. The redder colors are obviously higher quantities of this stuff, and these are determined by a digital drop PCR measurement. So this is a molecular biological test on total mass of material present in DNA. You can see we get spatial variations in each of these maps, and you can see there are different, this is the different substrates. These maps show us the spatial distribution, and then this is the bulk water, and you've got to be careful, this is comparing quantity per volume, and this is quantity per unit area of biofilm. But you can see that we already get a map of the occurrence of the Mycobacterium, and we can do more. We can then start to quantify how this relates to the level of disinfectant and so on in the system. I should point out that one of the most troublesome areas for us was down here in Sentosa, where at the time we did this study, water age was very high and there was very little residual disinfectant. Once you've looked at one or two of these species, you realize you've got to look at the whole community to understand the microbial ecology, and this becomes a hugely challenging topic, and I'm by no means a microbiologist. We did some tests jointly with CELSI, and I see Stefan Wirtz is here. So we did our first set of tests on these things, and this is just a summary of some of the key observations. You can say we've got a sample from one zone where the water age is low, meaning we have high disinfectant present, and then you can see we have a second zone of high water age, very low disinfectant level, and you can see we're comparing the biofilm on stainless steel versus the bulk water. And the key thing to take away from this diagram is the similarity of the color distributions in the diagram, and you'll realize that Mycobacterium, very strongly present in both the water and the biofilm forming. So these are in pipes very close to the supplied water with a very high amount of disinfectant present. The color over here, Nitrospira, is a nitrite oxidizing bacterium, and you can see it's heavily present in the high water age. So we're nitrifying water in this area, which is a very bad thing. And I'm sure that operators of water systems are aware of these things indirectly through other measurements, but this is essentially giving us a clear indication of the relationship between the water and the biofilm. And we know that Sentosa, they've done a lot to improve water quality, so this is not a giant surprise. So what are we doing since? Well, I just want to show you three pieces of work that we're involved in. One is essentially trying to characterize bacteria in drinking water, looking at the microbial ecology, extending this study. We also are doing quite a lot of work these days to just quantify total bacterial activity, and I'll show you, we're using a very simple tool called Bactiquant, which is a measure of enzymatic activity and links to the total cell count, and it compares very favorably with much more expensive flow cytometry. Then we're looking at online measurements, principally with this Alvimprove or a modified version of it, where we're looking at bioelectrochemical signals. And we're validating what we're seeing here in the lab using confocal imaging, where we can actually see the biofilm growth and relate it to what we're measuring. And then the third piece I'm just going to show you is critically important. We now have research networks that everybody can use here on the NTU and NUS campuses. So our problems on what I described previously were that PUB owns the operational water network and all the sensors, and none of the data are accessible to researchers. They're all essentially under lock and key, and access to the water network is very much restricted. So we've had to build our own research test beds here in order to pursue this work over a long term. And we've built these two research test beds on NTU and NUS campuses over the last year. They're now just operational. This is the situation at NUS. This has been a win-win situation for the campus facilities management. Both universities were losing water from leaks and bursts, and they were very happy when we came along and installed sensors in the network, and they're now paying for more of them. So this has been very good. Visenti, the company we formed from the first phase, manages the network, and the data, as I say, are accessible. In fact, the sites are accessible for measurements, which is great. Some of the data we've got, before we get to the network, I just wanted to show you that we've been looking at the microbial communities, the abundance, first of all in pipe samples. So this is when we install the sensors, we extract pieces of the pipe so we can see the abundance on the surfaces of the pipes that exist. And we've already started to establish that there are cyanobacteria in some of the pipes. There are opportunistic pathogens, at least in one of the 3 locations here. And we've found nitrospira, so we've found nitrifying bacteria. So we've got some interesting actors in the communities in these systems. Spare you this. The Bactiquant test, this is a sort of in situ, well, it's a test that you can do in situ. In essence, you apply an enzyme substrate to your bacteria, which creates a fluorescing compound, and you measure the intensity of the fluorescence. Gives you a measure of the total count. What's interesting about this is the Bactiquant measurement, which is done almost instantaneously, is almost linearly correlated. This is on a log, it's log linear. So it's a log of the Bactiquant versus the log of the cell count, so it's a power law function, correlated with the cytometry that would take a lot longer and cost a lot more money. So it gives you a good index of total cell count. We know that drinking water in general, if you take the tap water around the campuses, the Bactiquant value is below 10 to the 2 in most cases. This is the baseline we see. And therefore, we've started to use this as an index of bacterial activity within the drinking water. And we're thinking now about whether that can move to an online detection form too. We've done a lot of work in the lab with bench scale reactors, where we're basically dechlorinating the water, we're removing the disinfectant, and then we're progressively testing with online sensors how biological activity develops over time, and in some cases we're spiking that activity. The results are very preliminary at this stage, but I just wanted to point out to you that the results are not obvious at all. So this, for example, is the BQ value, the bacterial activity in water. The blue is unspiked water, so this is the water which is dechlorinated and left over an extended period of time. And then you can see the other 2 tanks where we've spiked the water, and you can obviously see a huge difference in the biological activity in the bulk water. If you swing across to the coupons, these are samples of the substrate we've put in, you can see it doesn't follow the same way at all. What forms in the biofilm doesn't correlate in the same progression as what occurs in the bulk water. So we're investigating in more detail this process. So you can say we've learned biofilm sample shares similar microbial communities as drinking water, but not developing in the same way, I suppose. And the Bactiquant is very useful. In the short time remaining, let me just give you a very quick overview of the Alvim-type probe. This probe is measuring essentially electron transfer across a surface when biofilm develops. The principle is if you have the potential against the current, different amounts of coverage of the surface with biofilm produce different responses. So this would be the fully covered surface versus the clean surface, if you like. The measurement works either in an intensiostatic mode, where you hold the current and you measure the change in potential, or you can, this is in fact what the commercial probe does, or you can hold the potential and measure the change in current as the surface gets covered. We spent an awful lot of time using the commercial sensor before we realized that it didn't work very well, and we've moved back to this potentiostatic mode. I'm going to spare you the detail of how the methodologies work, because we're out of time. I just wanted to show you the potentiostatic result, where we're measuring the signal as the biofilm is forming on the surface, and we're correlating it with measurements on the coupons that we see. These are on millimeter square coupons, and you can measure the green are essentially the live biofilm, the red are dead, and the yellow is something which has been live and is now dead. And we can measure the amounts of live coverage over the surface and correlate to that signal. So we're learning how biofilm forms on these surfaces of water pipes for different substrates. And the next step we're planning is to move this potentiostatic probe out into the test bed that we have on the NUS or NTU campus. I think I'll spare you the rest of the detail. But I would encourage you to contact me if you're interested in learning about, interacting, getting data from, or developing research associated with these networks on the NUS or NTU campuses. Thank you very much for your attention.
Time for a question while we're switching speaker, and otherwise Andrew will be part of our panel at the end of the session.
Yes. Yes. Yeah.
In which the horizontal axis was different species and their abundance, if you like, on the other axis. I'm wondering with what pathology each of those are associated, and what are the limits that, for example, the regulations in the United States imply?
Well, the regulations tend to relate to things like E. coli and other indicator species, as you know from Janelle's work here. So that's historically how people look at bacterial. I don't think much is known about the bacterial communities inside water pipes, so there is no regulation of this per se. There's also no measurement of it critically. That's the thing I was trying to drive at. So what we're seeing here is a way of quantifying growth of bacterial communities inside the pipes, and we're trying to understand what the ecology looks like and how it's related to the way you treat the water. That's the way we're approaching the problem.
Okay, our next speaker introduces himself on his first slide. We welcome back Asensum alum Masaki Kitajima, who is now, as shown on the slide, an assistant professor in the Department of Environmental Engineering at Hokkaido University, and he'll tell us how to detect viral contaminants in water. So Masaki, welcome back.
Thank you, Les. Can you hear me? Okay. So today I will be talking about our research on the development of AHTA sensor technologies for rapid virus detection in water. This research was initiated in 2015 as a Sensum Research Initiative project and has been carried out in my group in Japan. So the primary target in our sensor development is norovirus. Norovirus is the major cause of foodborne and waterborne gastroenteritis worldwide, and also known as an important viral contaminant in water. In fact, norovirus has been detected at a few points in Singapore's urban water cycle, specifically in untreated and treated wastewater and surface waters, according to the reports from Singapore. So these results have been obtained using polymerase chain reaction PCR-based methods, which are widely used for the detection of viruses in the environment. However, it takes at least a few hours to obtain the results and has limited on-site application. What we are proposing in this work is the use of a biosensor. A biosensor rapidly converts a biological response into a measurable signal. A biosensor consists of two major components, a recognition element and transducer. We use aptamer as a recognition element and electrochemistry or colorimetric measurement as a means of transduction. Now, what is aptamer? Aptamer is a synthetic functional DNA or RNA that falls into three-dimensional conformations binding to a specific target. This is an example of Aptamer binding to norovirus protein. Aptamer offers many advantages as a recognition element of biosensor. First, its high binding affinity. The affinity is comparable to or even higher than antigen-antibody reaction. It is highly specific because it recognizes unique surface structure of the target. Its high thermal stability makes it robust against ambient temperature variation and reusable with regeneration by reversible heat denaturation. It can be produced via in vitro chemical synthesis, which provides high purity and constant quality with low production cost. Lastly, we can easily attach chemical modification to an aptamer for various purposes, including immobilization and labeling. So this is the framework of our research. First, we developed a MEMS-based electrochemical aptasensor. This was done as a Sensum Research Initiative project in 2015, as I mentioned in the beginning of this presentation. Then, after I moved to Japan, we've been carrying out the research on detailed electrochemical analysis of aptasensor performance and optimization. These two parts are on electrochemical aptasensor. The third part is on the application of colorimetric sensor using gold nanoparticles, which is also an ongoing research in collaboration with other researchers in Hokkaido University. We are still at the phase of proof of concept towards the development of these technologies for norovirus detection. So we used mouse norovirus as a model virus, and mouse norovirus-specific DNA called AG3, DNA aptamer called AG3, reported in the previous research. So first, let me start talking about the development of a MEMS-based electrochemical aptasensor for norovirus detection, which was carried out in collaboration with Professor Jianmi Miao's group at Sensum and NTU. In the development of electrochemical sensor, we used a standard benchtop electrochemical workstation, as well as a portable electrochemical measurement system called WeStat. And the WeStat is as small as credit card and offered with a reasonable price, only around $130. We perform two types of voltammetric analysis: cyclic voltammetry, CV, for characterization of surface properties of electrode, and square wave voltammetry, SWB, for virus detection. For the development of aptamer MEMS device, we immobilized thiolated aptamer on the gold electrode through the affinity between thiol and gold. These figures show CV profiles and before and after aptamer immobilization. As you can see here, aptamer immobilization on the gold electrode surface substantially reduced the peak current, which indicates that aptamer monolayer, self-assembled monolayer, was formed on the gold electrode surface. The WeStat showed CV profiles similar to the benchtop workstation. Then I'm going to show you the result of virus detection by aptasensor. This figure shows SWB profile, and this line shows SWB profile for buffer without virus. When 500 PFU of virus was added into this buffer, we observed significant decrease in the current intensity due to the binding of the virus to the immobilized aptamer. Again, the WeStat showed SWB profiles similar to the benchtop workstation. Here I summarize the key outcomes of our IRRI project. First, we fabricated novel MEMS aptamer device. We confirmed aptamer immobilization by electrochemical measurement as well as visual observation of labeled aptamer on the electrode surface. Second, we evaluated mini electrochemical system, VStat. By combining this MEMS optimal device with VStat, we can potentially develop a portable optosensor platform. Lastly, we demonstrated potential ability for quantification because our experimental results showed clear titer dependence response against virus inoculated in this system. So these results have been published and described in this paper already, published in last year. Next, so this is the ongoing research in my group at Hokkaido University. So we are working on detailed electrochemical analysis of sensor performance and optimization. We started from re-evaluating the conditions used in a previous study, reporting the first development of norovirus optosensor. Specifically, we evaluated redox species used as a probe in electrochemical measurement. We also evaluated electrode material, gold nanoparticle screen-printed carbon electrode used in this study. And we used screen-printed gold electrode. Lastly, we evaluated virus detectability of this sensor in pure water and tap water, and we determined response time. First, redox species, evaluation of redox species. We carried out electrochemical measurements with gold nanoparticle carbon electrode in 4 types of buffers. Electrolysis buffer without probe, without redox species, electrolysis buffer with ferricyanide or ruthenium only, or with both ferricyanide and ruthenium. This figure shows CV profile, and the color of the lines corresponds to the color of the text of buffer composition. These 2 peaks are derived from unique redox potential of iron and ruthenium ions. As you can see here, largest peak current was obtained from electrolysis buffer with ferricyanide only, indicated with blue line, which demonstrates that this buffer composition is the most suitable buffer composition. Additional advantage of not using ruthenium ion is that we can reduce the cost because ruthenium is a rare metal and expensive. Second, we evaluated electrode material using the buffer condition judged to be the most suitable in the previous slide. We used gold electrode, gold nanoparticle carbon electrode, as well as carbon electrode without modification as a reference, without and with aptamer. The color scheme is the same for SWB and this table. Again, we observed a clear peak here derived from iron ion. So we determined relative current intensity, which means the peak current intensity relative to the baseline, and compared different electrode materials. What we observed here is, first, we observed similar profiles from gold nanoparticle carbon electrode and non-modified carbon electrode, which means that the reaction on the gold nanoparticle carbon electrode, the reaction on the carbon material for gold nanoparticle carbon electrode is dominant for this electrode. Second, we observed highest peak current excuse me, from carbon, from gold electrode after optimal immobilization. So, sorry, excuse me. So we judged that gold electrode is the most suitable gold electrode material. Then we assessed virus detectability using the conditions judged to be the most suitable in the last 2 slides, buffer probe condition and electrode material. So these figures show SWB profile in pure water and tap water with different amount of virus input. As you can see here, we observe reduced SWB peak current when virus was spiked in both in pure water and tap water. And it was repeatedly observed that this response was observed within 1 minute. Here I summarize the key outcomes of the part 2. So we optimize conditions. Optimized conditions are for redox species, ferricyanide only. And for electrode material, we judged that a gold material, electrode was more suitable than gold nanoparticle carbon electrode, which was used in the previous study. And we determined the virus response time, and it was within 1 minute. The third part is on the application of a colorimetric aftersensor using gold nanoparticles to norovirus detection. The sensing mechanism contains aptamer and gold nanoparticles. In the absence of target, the aptamer is absorbed on gold nanoparticles, which maintains dispersion of gold nanoparticles, even at a high concentration of sodium chloride. The dispersed gold nanoparticles shows red color, whose absorbance peaks at 520 nanometers. However, in the presence of target, the aptamer binds to the target to form aptamer-target complex, and aptamer cannot be absorbed on the gold nanoparticles. Bare gold nanoparticles tend to form aggregates, even at a high concentration of sodium chloride. So this changes color of the gold nanoparticles from red to blue, which decreases the absorbance at 520 nanometer and increases absorbance at 650 nanometers. Based on this principle, we recorded the absorbance ratio A650 over A120, 520, to indicate the aggregation degree of gold nanoparticles. This figure shows the absorbance spectra of gold nanoparticle in pure water with different amount of virus particles. In this figure, you can see only a subtle difference. However, if you plot the absorbance ratio against the virus input, you can see a clear titer-dependent response. We observed similar response for artificially contaminated groundwater and filtered river water samples. So this is a summary of key outcomes of the third part. We evaluated gold nanoparticle-based colorimetric aftersensor as a novel virus detection method. We showed its applicability to virus detection, and our ongoing efforts include optimization of detection condition and parameters. And then we demonstrated potential applicability of the colorimetric aftersensor to environmental samples. In summary, we exploited electrochemical and colorimetric aftersensor technologies for rapid norovirus detection. And these technologies are simple enough to allow for on-site norovirus detection without specialized training. I would like to acknowledge the Sensum Research Initiative Program, which provided the opportunity to conceive this research and financial support to initiate this project. I also like to thank my colleagues from Smart Sensum, NTU and NUS, as well as Hokkaido University and National Institute for Materials Science in Japan. Thank you very much for your kind attention.
One question for Masaki.
Thank you. Yeah, eventually, I'd like to develop a method that can detect norovirus particles without concentration. But at the moment, I'm thinking of concentrating the norovirus first, concentrating norovirus particles first, to increase the chance of detection using this sensor before we show the validity of this sensor for detection of indigenous norovirus particles in water samples. But in the future, yes, as you mentioned, we will try to develop a method that can detect virus without concentration in upstream. Thank you.
Okay, well, thank you, Masaki. Our next speaker is Sensem's own Janelle Thompson, also Associate Director of SMART and a water quality expert who's been working in Singapore now for some time. And as her slide shows, she's going to talk to us about analyzing microbial water quality in tropical waters, I guess with some emphasis on Singapore. So, Janelle.
Great, thanks, Les. Good morning, everyone. So, as my title slide suggests, my Sensam research is primarily focused on microbial water quality, and we care about water quality for a number of issues. I've picked two pictures here to illustrate these. This is my little girl, Ella, and she's got her face in the fountain at the Botanic Gardens. So we care about water quality from the perspective of its safety for drinking and its safety for human contact. We also use water for a variety of applications in agriculture, in industry, and recreation. So there are many reasons we care about water quality, and shown here are a group of MIT students who are kayaking on Makritchie Reservoir, illustrating one of the reasons why we care about water quality in Singapore's reservoirs. So Singapore is a nation that is dominated by catchment area. 2/3 of the landmass is catchment, and this catchment is drained by approximately 7,000 kilometers of waterways that feed into 17 reservoirs that are situated throughout Singapore. And let's see, can you hear me if I'm over here? Okay. So these waterways and reservoirs are carefully managed by the Public Utilities Board, and the governing principle that they've put forward is the ABC program, and this stands for Active, Beautiful, and Clean. The waters need to be safe for recreational use, active. They need to be beautiful, so aesthetically pleasing, both in terms of visual assessment as well as the smells and even the flavor. And importantly, they need to be clean. They need to be safe for human consumption and human use. So my research at Sensam has really been organized into three major thrusts. First is evaluating methods for tracking sewage contamination in the catchments. Second, we focused on looking at microbial activities in the reservoirs that can affect water quality. And third, more recently, we've started looking at the potential for top-down control of these populations by viral predators. So I'm going to walk you through some of the questions and the results that have motivated our research in these three directions. Now, just by way of overview, I'll let you know that my training is as an environmental microbiologist, and the methods that my group primarily uses are focused on extraction of environmental nucleic acids. So we collect samples from the environment, we purify DNA or RNA, and in some cases we culture cells from the environment. And then we use these samples for subsequent analyses, such as using next-generation sequencing to generate to regenerate, I should say, the information content of the genetic material within the environment. And so from that sequence data, we can understand the diversity of microbes that are present in the sample. We can identify the microbes present in the sample, and in some cases, we can design methods to quantify the abundance of the microbes in the sample using that information. And if we focus on analysis of the genome content or the expressed genes, we can also gain insights into the activities that the organisms are carrying out in the environment. All right, so I joined Sensam right around the 2-year mark of phase 1. And I teamed up with PIs Pete Shanahan and Lloyd Chua, who were PIs in Sensam Phase 1, and they were looking at the issue of how to track sewage contamination in Singapore's catchments. Now, this is a little bit problematic because in the tropics, the traditional methods that we use in temperate environments don't work quite as well. I'm sure many of you have heard me talk before on this topic, but basically, in the temperate environments, we use E. coli as a marker for whether or not a water body is contaminated with sewage. In the tropics, E. coli can grow. So E. coli likes to grow at human body temperatures about 37 C, and we typically see temperatures in the range of around 30 C, which is perfectly permissible for E. coli's growth. So Pete and Lloyd and the students that they were mentoring at the time carried out work to look at the abundance of E. coli throughout the catchment and waterways, and they saw that the abundance was actually correlated to human activity. But they were recovering surprisingly high concentrations that were on the order of 1, 2, even 3 orders of magnitude higher than what you would expect in the US, higher than the GRAB sample limits in the US. So when I joined the team, I suggested we look at an alternative marker that targets an anaerobic group of bacteria that can't grow under oxygenated conditions, and that are thought to be symbionts of the human gastrointestinal tract, and therefore highly specific for human contamination. And so we selected the HF183 marker, which targets this Bacteroidetes organism, and at the time, this was kind of the best standard for considering these Bacteroidetes. And this marker is actually considered or used by the US EPA now for use in the United States. We saw that there was actually widespread detection of this marker throughout Singapore. So that was a surprising result, and it made us think whether or not this was actually a suitable marker for Singapore. I mentioned this marker had been developed for the temperate environments, and we asked the question, well, is this really a suitable marker for the tropics? So to address this question, we carried out a follow-on study where we took the samples where we had seen high levels of fecal indicator bacteria, including the HF183 marker, and we applied next-generation sequencing to determine whether there was a relationship between high levels of these markers and the proportion of sequences that were pathogen-like. So this runs you through kind of the details of the analysis. We sampled 18 sites, 2 different monsoon seasons, and we used next-generation sequencing to recover information from this 16S ribosomal RNA gene, which all bacteria have. It's a good marker for biodiversity. And we generated 1.2 million sequences. We applied algorithms for taxonomic classification of these sequences, which let us identify the bacteria at the level of approximately between genus and species. And then we categorized these into their similarity to known human pathogens, and we used the National Institutes of Health curated databases for that. We also classified whether our sequences were similar to bacteria that had been found in sewage samples in previous studies and in 2 samples that we characterized from Singapore. What we found from this analysis was that when we consider HF183, E. coli, land use, and collection month as potential predictor variables, a model of beta regression, which lets you model a distribution over an interval from 0 to 1, this model showed that E. coli was actually the best predictor for the proportion of sequences from pathogens or sewage-like taxa. And in fact, that HF183 wasn't a very good predictor for pathogen-like sequences, but it did predict the abundance of sewage-associated sequences. So this study just came out. This was part of the work of Jean-Pierre Nshimiyimana, who was a SMA3 fellow here at Sensam. And this was part of his thesis, and we've just published that. So our kind of initial motivating question, whether fecal indicator bacteria, in particular E. coli, was predictive for the distribution of pathogens, shows that in fact there is some information gained by looking at E. coli levels in the tropics. Even though the levels are very high, they still do seem to reflect the abundance or predict the abundance of pathogen-like sequences. So we can ask whether this is truly a proxy for sewage, or perhaps it's a proxy for similar environmental dynamics between the bacteria indicators and pathogen-like bacteria in the environment. So ongoing is a follow-on study using a second fecal indicator bacteria, Enterococcus. But I think this work underscores further need for reliable methods to identify human sewage contamination in the tropics. And this motivates our next study, which JP has done in collaboration with his advisor at the Singapore Center for Environmental Life Sciences, Stefan Wirtz. You're going to be hearing from Stefan next. And in this study, we asked whether tests for the human-specific Bacteroidetes marker could discriminate human from other fecal sources in Singapore. And in this project, JP collected about 300 samples, and these are samples from human volunteers, and then samples from a variety of animals, including some that are with particularly local flavor, like mynah birds and wild boars. So what he asked were, do fecal samples from humans, can they be distinguished from the fecal samples from animals? And he asked this question using several different tests targeting different Bacteroidetes markers, including the HF183 marker that we had studied previously, but also including some additional markers that had been developed and validated more recently. And again, I should emphasize that all of these markers are developed for temperate context, and really their application in tropical environments is understudied, and we just don't know how well they work in the tropics. Okay, so what we found was that the HF183 marker was actually one of the worst in terms of its sensitivity and its specificity. We did find, however, that two markers worked quite well in this data set. And so we've put this forward via a publication that's just coming out in Water Research as a suitable marker for use in Singapore. And so now this is available for ongoing water quality monitoring work. Now, in addition, we've also evaluated an emerging technology called digital droplet PCR, which is kind of an advanced method for quantitative PCR analysis. And we've shown marginal increases in the sensitivity and specificity of these methods. And this will be communicated via a forthcoming publication. Our feelings on this subject were that the improvement perhaps isn't enough to justify the current cost of the method. So this is an interesting conversation to have. And actually, I would suggest any of you who are interested stop by JP's poster, which is downstairs. Now, thirdly, we're interested in understanding how long these markers persist in the environment. So when you see them, does it mean that the sewage contamination is recent, or has it been there for a while, potentially? And this work is still in the process of being analyzed and formulated as a manuscript, but this is a picture that shows JP hard at work preparing his mesocosm samples, where he's looking specifically at whether these markers persist for long times in sediments. And one of the reassuring things we found was that, in fact, the Bacteroidetes markers don't persist that long. So if you see them, it indicates that there's been relatively recent contamination. All right, so the second major focus of my work is, again, on elucidating microbial activities in waters that can affect water quality. And so here you see depicted a picture of Kranji Reservoir. It's a really lovely picture. Note the lovely shade of green there. So this is algae, cyanobacteria. And it shouldn't be too surprising that Singapore's reservoirs have recurrent algal bloom problems because, like I said, these waterways, two-thirds of the catchment, or two-thirds of the landmass of Singapore is catchment. And it's a primarily urban environment, and so there's going to be things like fertilizer and traffic runoff that get integrated by rainfall and delivered to these reservoirs. And so it's a very understandable challenge to have, and you can see these blooms here. Here's a cyanobacteria concentrated in a plankton net. This is a surface scum that I took with my iPhone, like standing above it, about 5 feet above it. So some of the problems associated with cyanobacterial blooms are the production of algal metabolites that can be toxic. And this motivated my first interest in these blooms, which I pursued during SenSam Phase 1. So we wanted to ask, what are the microbial activities occurring in the blooms, and in particular, can we see toxin gene expression? So on the heels of the work that I was doing with Pete Shanahan and Lloyd Chua, we also carried out a overnight sampling of the Kranji Reservoir. So we literally got off the plane from MIT, took a quick nap, and then headed into the field for this. We collected samples over a 24-hour cycle. We extracted messenger RNA, used next-generation sequencing to determine what genes were expressed in these samples. And what we saw was that, as we could visually tell by the scums on the water, the majority of those sequences came from cyanobacteria, in particular from Microcystis, which is a known toxin producer. When we looked for genes involved in toxin production, we saw that they were expressed in all the samples. And this was actually the first time anyone had looked into an environmental sample and verified that the genes were expressed in situ, so in the environment. Previous studies had looked at cultures in the lab, shown gene expression, but this was the first time we had actually seen the genes being expressed in the environment. And we noted that the sequences were higher at night than during the day, and we hypothesized that this could be due to a differential predation pressure. Okay, so now we have all this sequence data. What do we do with it? Well, this is an example of one of the analyses we used where we took a reference genome from a sequenced Microcystis aeruginosa strain, and then we compared the sequence that we got from the environment to that reference genome. Now, the reference genome is useful because the genes in it are all annotated with fairly high confidence. And so what we could do was determine what sorts of functions were being expressed in the reservoir. So this is just a list of the top 50 or so most highly expressed genes. And we can see that these functions, the highly expressed functions, correspond to things like buoyancy regulation, photosynthesis, DNA repair. And all of these things make sense within the context of a cyanobacterial bloom, where the cyanobacteria need to float on the water to effectively absorb solar radiation so that they can photosynthesize. So we asked the question, what are the activities of the microbial populations in a persistent cyanobacterial bloom? And we saw high expression of activities that made good sense to us, buoyancy, photosynthesis, DNA repair, and production of pili, which allow the scums to form. We also saw that between day and night, we could see differential expression of activities for photosynthesis and, surprisingly, for toxin production. And further, analysis of the other populations in the reservoir suggested an interplay between the coexisting populations. So cyanobacteria expressed a wide range of transporters for nitrogen uptake, indicating that they could use things like urea, which is commonly used as a fertilizer. And Proteobacterial phylum were enriched in pathways for biodegradation, and it's been suggested in other studies that proteobacteria might be key players in degradation of cyanotoxins and cyanobacterial metabolites, so this was interesting. And we also saw that the transporter profiles of the different coexisting taxa indicated some level of specialization. So some taxa seem to be more keen or attuned to metabolizing sugars, while others seem to be more protein specialists based on their transporter profiles. Okay, so with this baseline information of the activities in the bloom, we also realized that we had there was another research group here in Singapore at NUS who were interested in similar questions, and so we shared our findings with them and we started to talk more about potential for collaboration. And this research group is a group of Professor Karina Jin at NUS, and this is her algal team here. And they were interested both in toxins, but also in metabolites that cause the water to taste bad. And this is a particular concern to PUB, because people are more likely to call and complain when the water's not tasting very good. So this is some data that Karina's lab had produced and shared with us kindly last year. If you look into the waterways, you can see concentrations of these two compounds, 2-MIB and geosmin. The threshold of human detection is about 20 for 2 MIB, and some typical concentrations from a 3-day study saw MIB concentrations up to 300. So you could detect this by smelling, makes the water smell musty. So we wanted to get a better handle on the populations responsible for making these off flavors. And we collaborated on a series of monitoring programs, both monthly monitoring and diel monitoring, where the metabolite concentrations were compared to the cyanobacterial and microbial diversity. And this is an analysis that was recently published in Environmental Science and Technology, where we showed that the water body studied here was co-dominated by proteobacteria and cyanobacteria with some actinobacteria, which can also produce off flavors and odors. And we saw that through multivariate statistics, we could see that the cyanobacterial populations were more correlated to the distribution of these off-flavor compounds than were the other types of bacteria in the water. So this supported the notion that the cyanobacteria are responsible for producing these off-flavor compounds. We're exploring this further with metagenomics, and that allows us to put together a metabolic model for the bacteria in the sample. And we are also exploring this with isolate-based genomics. And our goal here is to figure out how exactly the microbes are making these off-flavor compounds. So this is an example of 2 strains that we just published genomes for. These both make 2 MIB. The one has a canonical pathway for producing 2 MIB by a known mechanism. The other lacks any genes for a canonical pathway, and so this raises the question, well, how is it making 2 MIB? Now, I'll just mention one more avenue of research that we're pursuing, and I'll invite you all to come down to our poster on this topic downstairs in the poster session. But we're also interested in how viruses regulate the abundance of microbial populations in the environment. And the main motivation here is that you can take a virus that infects a cell, and under the right conditions, you could potentially use that to kill off a bloom. And our idea here is that instead of introducing a new virus to the reservoir, can we just stimulate the activity of the existing viruses? And so therefore, we're not dramatically altering the ecology. Rather, we're just helping these cyanobacteria basically to get a cold and drop down in population size. So we've done a couple of baseline characterizations for viral metagenomes in the reservoirs, and we have some work ongoing in the marine environment, and we're actually very excited to get the sequencing data pretty soon. And so please join us at the poster session. We can talk some more about viruses in the environment and other environmental microbiology topics. I'd like to thank my collaborators and the staff that I've worked with at CENSAM. CENSAM alums are Dr. Kevin Penn, Boon-Fei Tan, Martin Tay, and Jia Wang, who have gone on to establish careers in industry and working with the Public Utilities Board. Currently, I'm working with 2 recent graduates of the SMA3 program, Lei-Ling and Jean-Pierre, as well as some great research engineers and postdoc Raji Kaushik, who has joined CENSAM relatively recently. And I'd like to acknowledge my collaborators, Professor Stefan Wirtz, Professor Karina Jin, Pete Shanahan, and Lloyd Chua. Thank you so much for your time. Thanks.
Oh, sorry, Masaki.
Janelle, thank you very much for sharing excellent data on the genomic indicators in water samples. My question is, could you briefly explain how are you going to link between the data on microbial source tracking markers and actual public health risks or infection risks in water bodies based on your study?
That's a great question. And actually, so public health epidemiology is a huge field. It's not my specialty. One of the main recommendations that came out of our recent ESNT paper was that we need to start adding next-generation sequencing-based approaches to these large-scale epidemiological studies where you look at risk of, you do prospective cohort studies of people who go swimming. They get you monitor how many gastrointestinal or respiratory illnesses they get, and then you relate that to the levels of certain markers that you've predefined, where we're suggesting that we start predefining the proportion of pathogen-like sequences as one of those markers. So, we're putting the idea forward in the literature. And I would be very interested to collaborate on such a study. The epidemiology part would need to be coordinated through a public health branch, though. Thank you.
Okay, thanks, Janelle. And our last speaker this session, and mentioned in at least one of the previous talks, is Professor Stefan Wirtz in Civil Environmental Engineering at NTU, now full-time after a split existence with UC Davis the last few years. He's also a research director for environmental engineering in CELSI, the Singapore Center for Environmental Life Science Engineering. And as the slide shows, he'll talk to us about the fate of pathogens in the urban water cycle. Welcome, Stefan.
Thank you very much, and thank you for the invitation. I'm very pleased to be here, and I start with an apology. I cannot stay the whole day because we are in the middle of running our summer course, our annual summer course, and I'm the course director this year. But I plan to be back for the party tonight, if that's okay. So I've been asked to say a little bit about CELSI. So CELSI is a mouthful. It stands for Singapore Center for Environmental Life Sciences Engineering. And we started in 2011. You see the mission statement here, essentially working on microbial communities in any environment, initially mostly environmental niches and later on also biomedical applications. And we were really inspired by the transition that has been going on for a number of years now from the traditional look in microbiology, the view that you have to study individual cells or individual populations of species to learn about their metabolism and how they function. And obviously that has served us very well. We've learned a tremendous amount of things about microbiology, but it's not really what's happening in the environment, is it? Rather, we have a series of consortia. We have a metabolism that is essentially more driven by community interactions than by interactions between populations of one particular strain. So this inspired CELSI, and we've been in existence now since 2011. And the structure of this institute, which is a center of excellence, there are 5 such centers funded by the Ministry of Education and National Research Foundation in Singapore. What you are looking at here is more the administrative structure. We have 4 clusters. One cluster is on environmental engineering, and you can see that the urban water cycle, that was not such a good idea. The urban water cycle features very prominently, like here, and this is what I want to talk about to you as well. The other cluster that might be of interest is called metaomics here, and metaomics means obviously sequencing, also metatranscriptomics, metabolomics, and we also provide a service. So if anybody wants to collaborate with us, we're very open to that. But if you just want to have some sequences analyzed and you don't want to send them to MIT for whatever reason, we do that as well. And the third cluster looks at fundamentals of how biofilms, which are communities, microbial communities growing at interfaces, how these biofilms might interact. So it is more a fundamental look. So you basically put together individual populations, and because you know them well, it is easier to deduce their interactions. And then the fourth cluster is the one that has developed around public health questions, including biomedical applications, essentially anything to do with disease. And then assisting those 4 clusters is a integrated analysis unit, which is run by Ron Williams at NUS, who is essentially a systems biologist, a big data person who helps us in various projects. So I collaborate very closely with him. And so last but not least, I do want to emphasize a sort of an anomaly, I suppose. Many people who know Singapore well shake their heads in disbelief. We actually work together between NTU and NUS. So our center was established with collaboration in mind. So we are stretched across the two universities, and the laundry list of departments and schools you see here is just an indication of who works with whom. Some of these people are more closely affiliated and some of them more loosely, but there is a lot of interaction between the two universities. So that should serve as introduction. And now I'd like to talk a little bit about some research we're doing and good planning to put me behind Janelle, because Janelle has already given you a very good reason why it would make sense to look at surface waters and recreational waters. So right here, you see a depiction of various outbreaks in recreational waters or caused by use of recreational waters. And the blue line, the blue line here is the one that refers to gastrointestinal diseases. And also that was already mentioned by Andrew in the context of drinking water. And if you take a closer look at that line, you'll see there is a slight increase over the years. So this is the US alone. Worldwide, the use of fresh water has increased tremendously and will continue to do so. And there is an increasing use of recreational waters as well. So even though drinking water is clearly the most important question, we also need to look at the importance of recreational waters. And the ABC program mentioned by Janelle is a reason why we're so interested in Singapore, in what that will actually entail in terms of risks of recreators. And so the work that I'd like to present here is largely the product of one dissertation, very recent dissertation defended by Nandini Shome. And for this question, I do like to ask simple questions, by the way. For this question, how do pathogens behave? What is the fate and transport phenomenon of pathogens in tropical waters? We chose a flume, a mesocosm flume of 2 meters. We ran these flumes at VKC, the Van Cleef Center, which at the time was still managed by NUS. Now it's managed by PUB. And we had to decide between the use of real pathogens or surrogates, and as you might envision, it would have been rather difficult to get permits to use real pathogens, and probably is not necessary either. So we chose surrogates, and then we worked with a combination of experimental analysis, but we combined that with a simple modeling approach as well. So the flumes, I already said that, are 2 meters long. There was a simple recirculation, partial recirculation, and we had replicates of these, because when you're looking at interactions in microbial communities, replication is very, very important. And in addition to that, we ran these flumes to establish a biofilm. We call that period conditioning period. And because these were exposed to light, so basically if you've ever been to VKC, you know it's covered, but it's open. So the predominant organisms that would grow in these flumes would have to be photosynthetic, so dominated by algae and cyanobacteria. And this is how it looked initially, and this is how it looked during the study. So it was a pretty dense biofilm, but rather heterogeneous. The organisms that we added are listed here. Pseudomonas aeruginosa is actually an opportunistic pathogen. It was isolated from the waterways. Enterococcus, you just heard, is a widely used indicator organism. And then we had 2 phages. So phages, these phages are bacteriophages. They do not harm humans, but they are also highly specific. So the phage needs a host in order to replicate. And this is a setup showing how we structured the experiments. We would run 2 parallel flumes. We would either treat them with surrogates or not, so a total of 4 flumes at a time, and we repeated these experiments several times. And then there were 2 flow conditions. One flow condition sort of representing the low flow conditions in Singapore, and the other one an annual average. And so the first simple question is, where do these pathogens go if we release them into our controlled system? Do they end up in the biofilm? And these are 2 slides that are a little busy, but I'd like you to simply focus on the colors. The first organism is Pseudomonas aeruginosa. Down here is Enterococcus. And then P22 is a phage, a bacteriophage that needs Salmonella to grow. Salmonella was not present in our system. We checked for that. So you can essentially you can look at P22 as an inert tracer. And so the blue line refers to viability counts. And as expected, viability goes down very quickly. The y-axis shows the concentration at time t divided by the initial concentration. However, except for here, in one flume where they totally disappeared, you can see that they are always detectable, viable counts, throughout the study. Of course, this is not just the original concentration, this is also detachment from the biofilm. And then the other colors refer to gene copies. So these organisms are not necessarily viable and infectious. Now let's take a look at the biofilm, and we see that within 24 hours, which was the first measurement we took, the target organisms found their way into the biofilm, and they stayed there over time. Once again, let's focus on the blue lines. So these are 2 replicate flumes in blue at relatively low levels, but nonetheless, they're there. Even the phages stay viable. And this answered the first question. We have a problem. The second question then was driven by the realization that it would be good to know what the mechanisms are. So why are these organisms there? What are the main driving factors? So the first question has to do with a simple modeling approach where we said, all right, if we account for the various mechanisms that are non-biological, how much of the fate and transport of the pathogens is explained using that approach? Oops, I seem to be jumping here. So just very briefly, we divided our system into 2 boundaries. The dashed blue line is the aqueous boundary, and the red line is the biofilm boundary. This is the flume right here. And then the rate of change of the concentration of any analyte or pathogen would be a function of the initial spike, so we only added one pulse, the feed that comes into the mixing tank, and the amount of material or analyte that would be detached from the biofilm. So all of these are inputs, and then the loss would be washout, settling, and decay. And you can do a similar mass balance for the biofilm. So let's see what happens. So just for Pseudomonas aeruginosa on the left, in P22, looking at the water phase, what we see is that there is a good good agreement with the prediction. You see the open symbols all refer to the model. But then there are occasional detections in the water that we cannot predict. And that's because of sloughing from the biofilm. So biofilm theory would readily account for that, but it's not something you can model easily. For P22, which I just told you is an inert tracer, there is excellent agreement with the model. So in other words, no biological mechanism, and you wouldn't expect to find one. So we know the surrogates can detect. So the question is, can detach. So the question is, how important is detachment? So if I run this model again, and very roughly simply ignore detachment, you see something like this. This would be the model if you don't account for detachment. So detachment from the biofilm is extremely important in the system. And so let's take a look at the biofilm then. In the biofilm, just P22 here, I said it can be cultivable, so these are infectious, or just DNA measurements, and we see perfect agreement with our prediction. The reason you don't see any measurements down here is that it's essentially below the detection limit. Does the same apply, or did the same apply to Pseudomonas? Indeed, it did not. So here are the two flumes, replicate flumes. We see that there is additional cell growth, more than we had predicted with the model. And we find that in the second flume, at the DNA level, we also see more DNA than we would have predicted based on growth. And this would indicate most likely that cells have entered a viable but non-cultural state, for which we did not measure. Thank you. All right, so then, so the third question was if whether hydrodynamics are important. So if ABC will lead to changes in the flow regime, does it affect the fate of pathogens, whether or not there is low flow or high flow? And this is a comparison for Pseudomonas aeruginosa. We already looked at this part in an earlier slide, and now compare it to the high flow. Instead of loss back into the water from the biofilm, here we find we actually see less in the water than expected. So where does it go? We also found that the 2-log reduction time, or T99 time in water, varied with flow rate. And if we drill a little deeper and consider growth that is not already accounted for in our model, what we find is that under low flow conditions, Pseudomonas aeruginosa will grow more in the biofilm than expected. At high flow, the opposite happens. And so what this means is that the organisms are preferentially that the nutrient development, that the nutrient concentration inside the biofilm is influenced by the flow conditions, which once again agrees with theory. And interesting for E. faecalis, something different is happening here. Low flow, we don't really see much disagreement with the model, but for the high flow, we find that there is more growth in the biofilm than we would have predicted. And this complicates things insofar as we cannot just say every organism behaves the same way. Every indicator or every pathogen that is bacterial will behave similarly. That is clearly not true. But however, I also want to admit that we had a little problem with Enterococcus because Enterococcus was present in the feedwater. So we had a constant low input of Enterococcus, and we do not know how viable those organisms were. Okay, so then to kind of finish the story, the last question is whether or not there would be an effect of these pathogens or surrogates on the microbial communities in the biofilm. Unfortunately, Janelle has already explained a lot about the methods one can use for that. So we used next-generation sequencing, we used metagenomic analysis to tease this apart. And what you are looking at here, this rather busy slide, is a visualization of an ordination approach called NMDS. We are trying to tease apart the two different conditions. The blue condition refers to the absence of surrogate. So each one of these symbols refers to a monitoring day, a flume. Just a monitoring day. And the green refers to the surrogate additions. The vectors refer to various OTUs or species that would have influenced this depiction in the ordination. So the numbers are not relevant here. And no matter whether we look at low flow or high flow, it is, I think, quite obvious that there was no effect of surrogates. And we also performed statistical analysis using two-way Perman-Over and related methods to prove that, and that's shown down here. So basically, the bottom line is, in terms of the prokaryotic condition, there was no influence, which means these organisms, although they entered into the biofilm, they coexist. But they coexist, but they do not invade as such. And so what does all of this mean? And I don't have time to tell you about all of the research in this dissertation. I think the major conclusion is that surrogates will end up in biofilms, and therefore it is very likely that pathogens will do the same. What does that mean? We don't really know, but you just saw the importance of detachment. So if you have an ever-increasing number of pathogens accumulating in biofilms, at some stage there could be a risk. And it's very difficult to actually run a risk assessment on that sort of information, because usually we run risk assessment based on what we measure in the water. So here we're going to look at the biofilm as a reservoir. And second, flow conditions will affect the fate and transport of these organisms, but it will most likely be very pathogen specific. And so the good news is that the communities are unlikely to be affected by these invasions, even if you use relatively high concentrations, as we did in our study. So then I'd like to acknowledge all the people who helped Nandini in this work. Without going through all the names, you can see that there was a big effort in her field team, and she got help from a number of folks who did work like install the camera to make sure she could take photos and so forth. So I hope you found that interesting, and thank you for your attention.
Okay, speakers up to the comfy chairs, please. Masaki, Andrew, we have a little panel session here to end this, so yes. Anyone can ask any of you questions. Well, this doesn't need my moderation. Any questions for any of the speakers?
So I'll start. I won't interrupt anyone. Hi. Okay. So, Masaki.
Yep.
I thought your result on the, I'm going to get it right. Was it the norovirus aptasensor or something else? Anyway, you showed a sensor result where you actually showed detection in environmental samples, and that made me so happy because oftentimes you see development of sensors and they work really well in deionized water, but you don't know how suitable they are for application in the environment. And so I was wondering, when you did those analyses, the slide was pretty brief, but it looked like you had as good of a model fit as your highly controlled system, which is a little bit surprising. I was wondering what, if any, challenges you encountered when you were using environmental samples, and if you are optimistic that kind of more challenging samples, it might work as well.
Yeah, thank you for your question, Janelle. So we evaluated the applicability of our sensor technologies to real environmental samples. Especially, we did intensive evaluation for colorimetric sensor. What we found was that the existence of ions, cations, especially cations in water samples inhibit the reaction of aptamers. So that inhibits our detection. But what we found was that the addition of EDTA to reduce the effect of cation helps us to increase the sensitivity. And reduce false negatives in the environmental samples. That's we found. And for other types of matrix, so we did that evaluation for, especially for groundwater samples, because groundwater samples tend to contain high amount of cations. But we are going to evaluate some false positive or false negatives derived from some environmental substances, including other environmental samples such as sewage, surface water, and so on.
Okay.
Yep. Thank you for your question.
Yes, so along the lines of biological interference, so sewage would have lots of different types of noro or closely related things to norovirus. So is that one of the next steps to look at the specificity aspects?
That's why, right. So the specificity of aptamer is governed or is affected by the design of the aptamer. So we haven't really developed aptamer that is truly specific to human norovirus. That is our future task. What we used was a model virus, mouse norovirus that does not infect humans at the moment because, as I mentioned in my presentation, we are still at the phase of proof of concept of this technology. So our future efforts will include the development of specific aptamer targeting norovirus, human norovirus particles. Thank you very much.
Well, while we're playing questions from the panel to other panelists, I was going to ask Stefan. I was very fascinated by your presentation on fate and transport of the pathogens between the bulk water and the flume in the biofilm. I wasn't very clear about the substrate where this biofilm was forming on the flume, and obviously, having seen a study like this, it seems like this is a prototype study. I was wondering if you can relate this to the problem we've got in our water pipes, and what sort of differences and things we ought to be looking out for.
Yeah, thanks. The decision was made to not introduce an additional phase, which would have been sediment. Previous work, also by Sanjay Swarup, who originally designed these flumes, involved a sediment phase. And so we decided not to do that, so to have the biofilm grow on the plastic surface. So any effect related to surface, type of surface, that wasn't part of the question, and I think you would have had a biofilm regardless of what surface you use. Absence of sediments, I think, is something we should remember. And by the way, most studies that look at flumes do not include sediment.
You want to ask the second question about how it relates to pipe flows?
Sorry. So I think obviously the flow conditions here are, we had two types of flows, not very high flow, both clearly in the laminar range, and I think there is a commonality between drinking water systems and these systems, which is what is the effect of rapidly changing flow. And so we see that in drinking water because you have stagnant regions, and then you have or stagnant times, and then you have flow taking off. And we didn't have the time to investigate what happens when you have an event such as one of our typical tropical rainstorms like yesterday. And I think that will obviously have a big effect on these biofilms. So I would expect runoff on the one hand, lots of runoff, of course, and second, lots of detachment. So there will be transport to other sites as well. I think that's probably as far as we can go. The organisms present in drinking water pipes are very different from what we see here.
Water quality in reservoirs. Obviously, pathogens depend on rainfall and runoff, both of which are quite variable in an environment like Singapore. How rapidly do measurements have to be taken and analyzed, and agencies be able to inform the public about the suitability of contact, given this variability? That might be a Janelle question, I'm not sure.
Stefan, I think you should answer the question about the kinetics. I will say from our surveys of the diversity of microorganisms in the catchment compared to the reservoirs, we see very different microbial communities, and that suggests to us that the reservoirs are reasonably efficient at removing pathogens. Like, they're not part of the reservoir ecology. You don't see pathogens growing in the reservoir. Those reservoirs are dominated by a distinct microbiota that is adapted to that freshwater environment. So there may be trace levels of the pathogens, but they don't really show up in our data sets.
I think there is a simple, a lazy answer to your question, but it's not to discredit the question, which of course is very relevant. The lazy answer is, no matter where you are in which country, for the next 48 hours after rainfall, don't recreate, because that's when you have the exposure that's been shown over and over again at bathing sites, and it's a common sense answer. I am more concerned about the long-term effects of these rainfalls, because I think of these biofilms as an additional reservoir. It's a sanctuary for any organisms that enter the biofilm and obviously coexist. So the fact that even viruses that have no host to replicate can just sit there and wait for their time to be detached. Human viruses will behave the very same way. And so it may even be that we cannot gauge the true extent of risk of these rainfalls by just looking at what's in the water at different times. We have to consider what's in the biofilm. So that's a potential risk. But to formulate a policy, what to do beyond my common sense answer, I think is difficult at this stage, because we are still at the discovery stage. The next phase would be to actually study biofilms in sediments in the waterways and see which organisms are there. I can ask a question. I can ask Andrew. So I was fascinated by the fact that you have essentially found a way to do research using the NUS and NTU water distribution networks. How easy do you think is it to ask questions that relate to the effect of biofilms versus organisms, communities that are passing through? So if you like, that come off the filter and then enter the distribution system and just wash out or come through, and that's what you see at the tap. Because some people think that's all that matters, and other people think biofilms matter, because that's where you can enrich pathogens and then you get detachment events which are unpredictable.
Right. Well, I mean, our network isn't going to answer the question. I mean, it's going to address the second one, right? It's going to talk about the importance of biofilms within the main pipe network. But this connection business at the tap is a whole different story. And there's another level too in Singapore, of course, because people store water in roof tanks. So there's a water storage element that we're not looking at. So we're really only looking at one phase of the process, I would say. But it is an interesting one, because more and more people eliminate things from the tap outwards, and the one that they know nothing about currently is the pipe network. So it's kind of the missing link in the chain. So that's why I think it's important.
Well, I have a question. So all of this is distressing. I mean, I've told people for years, Singapore is the one place in Southeast Asia where you can drink the water out of the tap, and visitors come from Hong Kong and they're shocked you're drinking water out of the tap. Where's the bottled water? Say, oh, water out of the tap is great. So what's the I mean, in recognizing that the discovery stage and so on that Stefan's talking about, what are long-term options? Is it detection at the tap of things that have come off? So Andrew's talking some about sensing that where one may say, okay, there's been some event that's loosened this stuff, but what about controlling a biofilm in pipes? Do we need some magic coating on stainless steel that will retard the formation of biofilms? Where could we be 20 years from now that may remove some of the risks that have been identified?
This sounds like a question for me. Well, first of all, drink the water. There's no problem. Hello? Press something? There's no problem with the water here in Singapore that we know of. In fact, the standards of the drinking water here in Singapore are much higher than almost anywhere. In our studies, Masaki and I looked at a comparison of quality of drinking water with respect to pathogens and things compared to the US and Europe, and the quality is very, very good. The risks are high intrinsically in Singapore, primarily because of the tropical environment and the temperature of the water in the pipes. So there are intrinsic risks here in Singapore. In terms of, we haven't really evaluated the risk. I mean, that's the point I'm trying to make. We don't really understand our pipe networks very well to understand where the risks are located. And that's really what we're engaged on with this biofilm growth rate. We still think this is sort of essentially the parameter that people need to understand in order to appreciate if you want to pick up Stefan's line of argument, the reservoir of potential bad actors that can come in. So if you can control biofilm growth rate, you're doing very well. Practical infrastructure issues relating to what you do. If you've got a serious problem in your water pipes and you cannot flush it out, it is a real nuisance. And every utility knows that. And it becomes a huge problem for them. I don't know if you follow in the news, these things are small news frequently, so pervasive news. New Orleans had a terrible problem with its water supply. A child died from, what was it? What's the amoeba called? Naegleria. And they spent 3 months flushing their water supply in this area. So it becomes enormously difficult to remove when you get things in there. So I think, other than ripping up pipes and things, it's very, very difficult. And obviously, antimicrobial coatings on pipes would be a wonderful thing. In fact, we talked very extensively to Karen Gleason, you'll recall, about work on just that process, that if you could coat water pipes with antimicrobial coatings, that would be a huge advantage. But it's just not a practical reality. It's going to happen anytime soon. But the potential certainly exists. So.
Can I give a short comment? One of the things we haven't really looked at is the nutrient levels in drinking water in Singapore. In theory, if there is no nutrient in drinking water pipes, there won't be biofilm growth because bacteria cannot grow without their food nutrient. For example, in the Netherlands, they are monitoring the nutrient levels or carbon contents in drinking water pipes to suppress or to minimize the growth of biofilms in the pipes. In Singapore, we have high temperature in the tropics, and this is favorable condition for pathogens and other microorganisms to grow in the pipes. So this is something we have to investigate and consider in the future to minimize biofilm growth in tropical environments in water pipes.
Thank you. Well, I didn't mean to have the last question, but we're, I think, at a point where we should stop. The speakers, you can nab them during coffee break, and I think our slightly, modestly late start to get a quorum will mean that we start our coffee break 20 minutes after schedule. We'll finish 20 minutes after schedule, so at 11:15, please be back here, and we'll start the second session. Let's thank the speakers again.